introduction and then talk about why you chose this project. Go.
Hi, so my name is Adrian Duraku, and my co-op is at Ford, and I am a ME major, senior ME major. And so one of the reasons I chose this project is because I have a focus in bioengineering, and essentially, for me, I've always loved microbiology. Microbiology is something I'm super passionate about, and me being able to at least dive somewhat into it, and also connect this concept to some of the more advanced mechanical engineering classes we've been experiencing so far is something that I thought would be really cool, a really good idea for a project. But yeah.
And I'm Alex Zhang. I'm a senior one here at Kettering University, and I'm in mechanical engineering. And I think viscosity would be pretty sweet in multiple liquids, and blood and egg whites being one of the best liquids there is in the world. I was just curious of how to do it for our presentation and our project.
All right, get started. So just a quick project summary. Essentially, our goal with this project was just to simply test the viscosity of blood, a non-Newtonian fluid, would change. Essentially, we wanted to see like how feasible this project would be, because we have very limited resources at Kettering, and so we tried to just basically scope out the project. And so another thing we also looked into was just testing our blood types, because both of us forgot, we were told when we were the doctors told us, so it was cool to go through that experience again. So we bought these on at home testing kits. And then and we did the end summary of this project is that we did, for a lot of the parameters we did test, we saw changes in viscosity. Not for blood, but for egg whites, which we'll get into later. So some of the more project goals, I was just kind of talking about this. Essentially, just basically wherever we could test blood, which ended up only being one parameter. We tested blood, and essentially we tested our blood types, and we just observed the change in viscosity. And we just learned more about the fluidity properties in general, mainly non-Newtonian fluids and viscosity non-Newtonian fluids connects directly with viscosity. But yeah, that is basically our main project goals. So now to get into the materials a little bit. So our materials ended up being around 7 egg white cartons, which is a lot, some blood lancelets, 2 different types of microscopes. So the reason we have 2 different types of microscopes there is we had access to the biology lab, but not full access, just due to the lab tech and our bio professor not having enough time. So what we ended up doing is we were able to get a few pictures with a heavy duty microscope, and the majority of the pictures we took was with a lower magnification portable microscope that we could test anywhere. We actually ended up collecting the majority of our data within the LC. So that was a fun experience. So then obviously also I mentioned earlier blood testing kit, which was this little thing that you would prick your finger. You get 4 droplets of your blood, and you would dip it into these chemical solutions that would react a certain way, and depending on the reaction, it would tell you your blood type. And if there was no reaction, you're O negative. So I'm O positive, so I'll get into that later. I was close, but we also had a rotary viscosity meter, which Alex will go into later. So now, more realizing the limitations of our project was blood was not going to work out as much as we hoped, and that was mainly because just due to the fact that we overestimated how much blood we could extract from the lancelets. We thought we could maybe get at least fill a beaker this size, but unfortunately, every time we tested the lancelets, we got barely a dropout. And another thing was the pain level was kind of high too. We underestimated how many nerves we have in our fingers. Yeah. So through research online, and also actually I made a post on Reddit. I told them about the project a little bit, and I asked well, what could we do as a substitute, or what would you guys recommend? And they told me egg whites. And I looked into it more, and I talked to my biology professor, and she said that's a good idea. It's pretty close, and the reason being is because it is a protein-dense fluid, similar to blood. It has many types of molecules in there that kind of interact with each other, and they're kind of jumbled together to form these more advanced proteins. But yep. so now to get into the more fluid mechanics side of things, which the first thing we would like to talk about is viscosity. And basically, what is viscosity? Viscosity is basically a fluid's level of resistance to a amount of shear stress. Basically, or in simpler terms, like how thick a fluid is. The more thick a fluid is, the more shear stress that is required to actually see it break or see it move. And at a more microscopic level, the molecules are more densely packed. Not densely packed enough to form a solid, but more form like something that gets closer to a gel. And obviously, less viscous fluids, molecules are more spread apart. And yeah, so sorry, just 1 second. Oh yeah, and also depending on the type of fluid, the viscosity can fluctuate very easily. Yep, you can go on. So now getting into our other fluid mechanics concept, which was non-Newtonian fluids. And essentially, a non-Newtonian fluid is a fluid that what's it called? That can change can fluctuate very easily. Its viscosity can fluctuate very easily depending on pressure or force, yes, as well. And basically, because of the uniqueness of this fluid and how easily it can change, that can kind of lead to problems in the industry if non-Newtonian fluids are required. And from what I've seen, they are sometimes avoided if possible. But there's some situations where they can't be avoided, and an example of that would be in the medical industry with blood. Blood is a non-Newtonian fluid that cannot be avoided in a lot of situations. So they have to figure out ways to pump blood into whatever they see fit. Which it is possible to pump a non-Newtonian fluid. It's just more parameters are required. Yep, you can go on here. Now to get into the more microbiology side of things. So basically there's a few things that will change the viscosity of blood. So here we have a picture of a low viscous blood where you can see the blood cells are more spread apart, and then the high viscosity blood, which the blood cells aren't bigger, they're just more closely packed together. And basically, and that kind of correlates with the idea of molecules being densely packed together instead of molecules at a more like at a larger level, we have blood cells, which function as those molecules. And also, just one more second. There is also, there is also, basically when more viscous blood is typically seen in typically means that there's just more blood cells all densely packed together, which can actually be detrimental or helpful depending on your situation in life. If you are a young person who exercises a lot, you will have typically a higher red blood cell count, and that's just due to your body requiring more oxygen, and that's not a bad thing. That actually means you're more healthy. But where it becomes an issue is older people with high red blood cells, red blood cell counts, because they will usually suffer from heart conditions and other organ conditions, which we'll get into later. But yeah, and that's it. You can go on. So now to get into the other microbiology concept that will also change blood viscosity. So basically coagulation. This is basically a concept that basically is a lot of the reason why life exists, because it's basically a counter to any type of cut or damage to your body can basically help stop that. So basically there are these little things called platelets in your bloodstream that flow throughout your blood, and platelets are the term for them are cell fragments, but they aren't alive. The better term, in my opinion, would be a more protein a protein kind of jumble, like a protein mix of a bunch of different just proteins and biological material. So essentially what happens here is when you get a cut or you have some sort of chemical reaction, that chemical reaction could be venom, your blood will, to start to artificially increase the thickness of it, what will happen is these platelets will trigger and they will start to form a mesh around your red blood cells. And when that happens, when they form that mesh, they close in on each other, and then that densely packs all the red blood cells together, and then in turn increasing viscosity. And then another thing to mention is genetic disorders are also increase your blood viscosity as well. So an example of this, a really good example, would be sickle cell. Sickle cell basically causes your the normal donut shape of your red blood cells to be instead formed like a sickle, which means that the people with sickle cell disorder, or with the sickle cell disorder, basically cannot. Their blood cells they still function, but they're not as they can't carry as much oxygen. Not only that, they actually, since they're not that donut shape, they tend to get like hooked on like the parts of the veins or to each other, and they've also been recorded to be more sticky. So that also increases your risk. Basically, people with this type of blood have very viscous blood, which is obviously, as we mentioned, could be detrimental. So now, getting to one of the first, our initial, this is our first parameter that we tested. And this was more something that, this was basically a scoping idea to see if blood would work, which this told us that it wouldn't work. But we did what we could. So after we tested our blood types, we discovered that I'm O positive and he is B positive, which and another way to talk about these blood types is that basically I can take, Alex can take my blood, but I can't take his blood. In terms of a donor. So that's due to the level of antibodies and antigens in the bloodstream. And yeah, so basically what we did is, so to still see some sort of reaction, what we ended up doing was we ended up just splitting our blood 50-50 and mixing it together and analyzing it under our microscope. So if you get into our next slide here. So before we get into the mixing of it, here is my blood under a microscope, which you can see the donut shapes. And basically, this is just, as soon as I took it out, we analyzed it on a microscope. And you can see, basically, this is how the blood should normally look, mostly. So here's so this is the same sample, but after a few minutes of letting the blood dry out. So you can see the very slight coagulation starting to occur. You can see the clumps right here. And what I really liked about this picture is you can actually see the blood cells stacking on top of each other, which is something I've never really seen before. You can really see the donut shapes in this one. So now to getting into our mixing scenario. So this is the O positive mixed with B positive. And essentially, you can see the cell walls are just super compact together. They are super just closely meshed. And this was almost immediately, we recorded this picture, we got this picture almost immediately as soon as we mixed our blood. And that's due to the fact, what essentially happens is the antigens will attack the antigens will attack the antibodies. And when that basically attacking occurs, it triggers your platelets it just triggers your platelets, and they form that mesh and they start artificially doing it, even though they're not supposed to. They do that in thinking that there's something wrong with the bloodstream. So that is why we're very careful on when you're donating blood, or else you could cause someone to have a heart attack artificially or a blood clot. But yeah, we can go to our next.
Now I will talk about the process for our experiment and our data collection. That we were trying to find for the change of viscosity. So we used the meter, or the rotor meter, and during that meter process, we had a rotor that spin counterclockwise at 60 rotations per minute, or RPMs. And using that, we found our measured results of temperature with degrees Celsius and viscosity at millipascals. And we found one control variable, and we also did 8 different tests with 5 different parameters, being temperature, dryness, exposure, dehydration, and different off-counter medicines and melatonin.
To simulate sleeping.
Yep.
For melatonin.
And yeah, and for each test we did 3 samples each, and we took the average of all the viscosities of all 3 examples, and we did all 3 samples at all different times throughout the experiment. So we did a control variable for, it's just standard normal egg whites, and the normal viscosity for the egg whites is 10.1 millipascals, or a temperature of 22.5 degrees Celsius, as you can see here from our table.
This is also one thing to note, is this is the handheld microscope under a microscope, so it's kind of harder to make it out, but you can see something there. So yeah.
Yep. as we get on the experiment too, you'll start to see that the more different parameters had different microscope pictures as well. So for our first parameter, we did temperature and how egg whites drown within the temperature that they're placed in the environment. We did 3 different tests. So we did a negative 20 degrees Celsius, a negative 80 degrees Celsius, and a 70 degrees Celsius, which is for hot, cold, and super cold. And
one thing I wanted to just mention is we tested at the temperature. Basically, it's supposed to simulate like a person, not obviously exposed to that high degree of temperature, but just close enough. We only exposed them for like about an hour to 30 minutes, and so just enough so we could see some sort of change and we can record that rapidly. But yeah.
yeah, so like Augian was saying, we placed the samples for an hour and we stored them in the negative 20 degrees Celsius freezer that the biolab gave us. And then we did another freezer which had negative 80 degrees Celsius, and it was a separate freezer that the biolab gave for us, but instead of placing it for an hour, we only placed it for 30 minutes, and we didn't realize it was super frozen for 30 minutes, so we had to let it melt out for another 30 minutes, and ice definitely played a huge factor as the rotor was hitting the ice when collecting the data from viscosity.
Yeah.
So
You can see here that it significantly increased the viscosity for the colder, yeah.
And then for our hot 70 degrees Celsius, we only placed it for 30 minutes, as I think the heat will definitely heat up the egg whites during 30 minutes, and it tends to be lower than our normal standard viscosity level for egg whites. So our second parameter was dehydration. So pretty much we wanted to see like how people get dehydrated without water. So we simply heated.
To simulate it.
Yeah, we basically simulated egg whites being heated for 60 seconds by placing a hair blower on the top of the egg whites, and you can see here from our results that we got an average of almost 9, but there was one where it was odd result.
This parameter was like it was more than we thought of when we were initially still planning on doing blood. We still decided to do it because we thought we could still simulate dehydration, which there might be some error here because there was a lot of open-ended. We just assumed that maybe, well, if we just blow the egg whites for a certain amount of time, then maybe that could start to dry out the egg whites. But yeah.
And then for our third parameter, we did melatonin, so pretty much just mixing the egg whites with melatonin, as we use 14.8 milliliters of melatonin, and it pretty much like we wanted to simulate this to see how like we sleep, and we as blood was one of the main factors that we want to use before we use egg whites. And then for our fourth parameter, we use 2 different medicines. So we use NyQuil, and we use called magnesium, which we just mix it similar to the melatonin, where we mix the medicine or off-the-counter medicines in there, and we wanted to see how like these medicines could affect the egg whites, similar to how it could affect us with blood. So we first used the NyQuil. As you can see here, we did the microscope picture. There's some bubbles that appeared during the egg whites, as we used 50 milliliters of NyQuil with the egg whites.
And there was an increase in viscosity with the NyQuil, yeah.
Yep. But there's definitely an increase of viscosity when we did the called magnesium, as we used 50 milliliters as well, similar to the NyQuil. And when we mixed it, it started bubbling up, similar to like how it popped with Mentos. So
yeah.
You can see all the bubbles there, and it was a huge mess. So we had to clean it up. And you can see from the microscope picture that there's a lot of bubbles. So, and it had a super high viscosity of like around 300. So.
Yeah, so definitely some error here.
And then our last parameter was exposure to the atmosphere. Pretty much we just left egg whites in 3 beakers for 3 sample tests for 24 hours, as we want to see like similar to blood, how like we keep it out there for 24 hours. And we just kept it at room temperature.
Decrease in viscosity too, yeah.
So overall in our results, we used 5 different parameters with 8 different tests, and our change in viscosities, the highest viscosity was the column magnesium with the bubbling, at like 305.1 millipascals, and our lowest viscosity was our temperature at the highest hottest level, which is 70 degrees Celsius at 6.7 millipascals. For our future recommendations, we wanted to change some times for trials, as I think some of our freezer and heater placement times were either too short, like, or too long, like an hour or 30 minutes. We feel we could have made it like 45 in the middle, and we want to maybe use different amount of liquids instead of just using egg whites and blood. But I know we had a limited time, so I know
Right.
that could also be a factor. And we also wanted to use different ways of measuring viscosity, because even though we did have the rotor meter, we were thinking of maybe using other stuff to see like if the viscosity read the same or got the same values. And yeah, and our final recommendation is just maybe investing in blood extractions or different tools with using blood.
Just for like the Kettering resources, because there wasn't much to measure viscosity here, and that's why we.
Mechanical engineering, yeah. That's what
Yeah.
we do.
And then also, this is more, I know that biology is starting to fade away at Kettering, but still, I would like to see some sort of like more stuff that you could interact with your own blood, because there's a lot of stuff you could, data and tests you could do on yourself, and a lot of lab experiments we could do.
Right.
I mean, in my biology class, we did one with like our spit, which I thought was a really cool one. But yeah. Just go on to our next slide here. So now to get into the relationship to environmental sustainability, this project is less about the environmental and more about the human side of things, and which I would consider relating to the environment. And essentially, if we further our research in the understanding of non-Newtonian fluids, that could definitely lead to just advancements in a lot of the medical technologies. Basically, like not even just blood. It could also extend to just different like types of medicines that also happen to be non-Newtonian fluids, or different types of fluids that we're dealing with that are still alive. But yeah. And then also, yeah, and then basically, this kind of research could lead to like basically just saving lives, because basically the bottom line is people with more viscous blood typically are at super risk of heart attacks, random blood clots. I mean, people die just getting a blood clot in their brain, and there's literally like that can happen instantly, and they don't even realize that because they're not paying attention to their own blood, which is something that a lot of people just they forget about, because they don't see it, they don't, and typically they don't feel it. So yeah. Now to get more into our personal project interest and just future recommendations. So we both mentioned, or we mentioned earlier, that we both have a really big interest on microbiology, because we're both bioengineering focuses, and basically it was really interesting just seeing the change in viscosity, just observing, because viscosity was something I knew of, but I wasn't really like super like. I wasn't super like well versed on it before I was interested in this project, but yeah, and then also basically for our future recommendations for this project, I mentioned this earlier. Just if there could be more resources dedicated to just the Kettering biology department, or also just the viscosity measuring, the viscosity rotary meters as well. That would be a great thing. And in terms of basically if this project should be repeated, we interpret it as like this our specific project, which I think it would be a great idea if we have access to those things, And plus, it would be really cool to maybe it wouldn't have to be like a full test, but maybe something that people could actually look at their own blood and measure the viscosity reliably. That would obviously that was another thing I forgot to mention was. Another reason we couldn't really test our own blood was because, as we saw in the second sample for the after a few minutes, the blood automatically, as soon as it leaves the body, your pressurized body, it just starts to coagulate instantly. So you need to have the anticoagulant. We didn't have access to that, but if this experiment were to be repeated, you would be able to test I feel like testing your own blood with the use of an anticoagulant could work. But yeah.
Another thing that I found interesting while learning about this experiment is that torque and shear stress can be used to discover viscosity, as I never knew just like the rotations could solve like for the viscosity within our experiment, and just measuring other liquids in egg whites. So we want to thank Dr. Berry, of course, for our lectures. Ray Russ for giving us the ideas.
Yeah, Ray Russ actually gave us the idea of finding a different model, like using a model instead of our actual blood. Because initially, I wasn't even sure about what to do instead of blood.
Yep.
And then obviously our biology professors as well. The lab technician was a big help. She dedicated some time, and yeah.
Yeah, we just also want to give special thanks to them for letting us use their labs
Yes.
as
Yeah.
well, because we spent a lot of time in there and we got a lot of good research.
Yes.
So,
Good.
so thank you guys. Any questions?
Any questions? One question.
So you decided to use egg white as a substitute because you could use human blood. If you did the experiment again, would it be worth considering any type of animal blood?
Animal blood? Well, that's also, I mean, that is possible, but it's, again, that comes into the price range. For this entire project, we basically, since we were kind of going off the grid here, we had to invest into ourselves. If this project were to be funded by Kettering officially, using animal blood I'd assume would be just as good. Maybe not exactly the same due to the fact, if we're looking for like the medical side of things, because there are slight differences in compared to animal blood, but yeah.
Yeah, Ray Russ actually gave us a suggestion of buying other blood, like a package of blood. So if we had more money, like Adrian said, we definitely would have tested that. So
Yeah.
yeah.
But just one more thing I wanted to mention. That would also, even with animal blood, that would still run into the same problems of we need an anticoagulant as well, and we need to pay attention to that because anticoagulants are kind of like, they're designed to keep the blood okay, but it's different even if it's different even outside the body, or especially when you interact with that, it's just it changes a lot of things. But yeah.
Okay. Any other questions? Okay, guys. All right. Yeah. Okay. Wow.
Cool. Sweet.
You guys have come like a long way
Yeah.
from that first proposal.
Yeah.
Yeah.
Wow. Well, it is about identifying issues and problems and coming up with the alternatives and solutions, and that is what engineering is all about. Now, in terms of the environmental sustainability,